Hi! In this lecture, we are going to explore some of the rules of gene expression. Now, in the last lecture, we learned about the processes that are involved in gene expression. Specifically, that we start with double-stranded DNA, and then that DNA is read by RNA polymerase to produce an RNA photocopy. And then that RNA photocopy finds a ribosome where it is translated into a growing polypeptide strand. Now, this two-step process can be broken down into each individual step, and each step has a name. Going from DNA to RNA, recall, is transcription. And from RNA to polypeptides is translation. Now, as a quick refresher, gene expression in a eukaryotic cell is separated by both space and time. And that is because the DNA is housed in the nucleus, a special compartment, where the ribosomes are out in the cytoplasm. So transcription is going to happen in the nucleus. And then that RNA must find its way out of the nucleus into the cytoplasm, where a ribosome will read it. In prokaryotic cells, there is no nucleus. Therefore, the DNA is not kept separate from the ribosomes. Consequently, both transcription and translation happen very close to each other. You might notice here that this orange line is the RNA, and it's not even done being transcribed, and ribosomes are already latched onto it and translating it. So gene expression in a prokaryotic cell is probably a little quicker than it is in a eukaryotic cell, in part for that reason. Now, the purpose of this lecture is to learn how to predict what the polypeptide strand will be if given a DNA sequence. So remember, DNA has 4 nitrogenous bases. They are A, C, G, and T. And if I have a particular sequence of DNA, let's say it's A, C, C, G, T, A, T, C, etc., can I predict what amino acid that DNA sequence will code for? And the answer is yes. Scientists have indeed elucidated those coding rules. So that's what we're going to get into today. The objective is to be able to predict what the RNA transcript looks like and what the primary sequence of the resulting polypeptide looks like if given a specific DNA sequence. So let's go ahead and get started. Okay, in the upper picture, I have part of a DNA double helix that's been stretched out a little bit. You'll notice that the base pairing rules still apply. A always pairs with T and G always pairs with C. Go ahead and take a look at the structure on the top and confirm that A is paired with T and C is paired with G. All right, now we are going to get into the RNA synthesis. So RNA is very similar to DNA, and in fact it shares 3 of the same bases, but one of the bases for RNA is different. So DNA bases are A, C, G, and T. RNA bases are also A, C, G, but there is no T, there's no thymine in RNA. There is only uracil. So uracil, or U, is substituted for thymine. So what does this mean? It means that any time I see a DNA base for A, I would normally pair it with a T if I was looking at DNA. But if I'm pairing it with RNA, there's no T, there's only U. So let's go ahead and erase this here real quick. And let's actually form the base pairing rules of DNA to RNA. So A is going to pair with U, C is going to pair with G like it always does, G is going to pair with C like it always does, and T is going to pair with A like it always does. So now we can use these rules to predict the RNA transcript that would be synthesized from this section of DNA. So let's go ahead and do that. Now notice that I've separated out the DNA a little bit more here, and now we have to pick which side we are going to transcribe. Turns out that one side of the DNA is left alone entirely, and the other side, called the template, is the one that actually provides the code that RNA polymerase reads from. For this class, you are not expected to be able to predict which side is the template and which side is the other side, but I do want you to be aware of the fact that typically RNA polymerase only reads one side. And in this example, it's going to be the bottom side. So RNA polymerase is going to come up here and we're going to start synthesizing RNA based on these base pairing rules. So remember, just rewrite them here real quick. A is going to pair with U, C is going to pair with G, G is going to pair with C, and T is going to pair with A. So I can use that to predict what this RNA is going to be. So I start here, I come up and I see I've got a T. What pairs with T? Looks like A. So I'll go ahead and put the A there. Now I move over to the next spot and I see A. And I come over here and say, okay, that pairs with U. And then the next spot, I see a C. So C is going to pair with G. And then I come over to the next spot. Again, I see another T. T is going to pair with A. I come over to the next spot, there's an A. So I look up at my rules and I see that A pairs with U. So why don't you go ahead and try and finish this using those base pairing rules that you see here. All right, if you did this successfully, you'll have something like this. So we have AUG, AUC, UCG. Now you might notice that this actually looks very, very similar to the strand that we ignored at the top. The only difference is that T is substituted with U. Okay. Now that we have this RNA, what are we going to do with it? We are going to send it to a ribosome, which through a sort of complicated set of steps is going to somehow read this RNA and translate it into the language of amino acids. So how does that work? Well, let's take a look at this picture here. I've copied our RNA strand down, and so what we see here is we see kind of a busy picture. I'll try to guide you through it. Let's start with our double-stranded DNA. So again, you see that C pairs with G, G pairs with C, T pairs with A, etc. And only one side is being read, and then you produce this RNA. And notice that the T's are missing, and instead of T's, we have U's. Okay, so we've already covered that. That's basically what I have here from the previous slide. So now look at what happens. It looks like if I follow these arrows down, it seems like 1, 2, 3 of the RNA bases somehow codes for one amino acid, and ARG stands for arginine. And then I have my next one is GGA. If I follow these gray lines down, that seems to code for yet a different amino acid. GLY is glycine. So if I'm looking here, I notice that it takes 3 bases to get me one amino acid. So 3 bases are read at a time to give me one amino acid. So notice here that I have 1, 2, 3, 4, 5, 6, 7, 8 amino acids. So to code for those 8 amino acids, if it takes 3 bases to get to an amino acid, then technically this should be 24 bases long. And if I went ahead and counted, you would see that indeed 8 times 3 is 24, and there are 24 bases in that RNA strand. Let's come over to our RNA strand that I copied from the previous section. How many bases are in that RNA strand? You might notice it is a replicate of 3. And so if I take a close look at this, I've got 3, followed by another 3, followed by another 3. So this section of RNA should code for 3 amino acids. 3 RNA bases that code for 1 amino acid are effectively known as codons. So this is a codon, AUG. AUC is also a codon, and UCG is also a codon. How do we know what AUG codes for? Or what about AUC? Or what about UCG? How do we know what any of those codes for? Well, it turns out that this has also been elucidated by scientists, and they have published a very handy-dandy translation chart, also known as a codon chart, that basically lists all 64 permutations of codons. There are 64 possibilities, and tells you what each codon translates to. So let's go ahead and take a look at that. Now, before you get too worried, this codon chart will be provided for you during any exam, so you won't need to memorize this, but you will need to be able to read it. So let's go ahead and try our section of RNA that I have shown at the bottom and see if we can figure out what amino acids these code for. The first one is AUG. So what I want to do is look on this chart and see if I can find AUG. And I find it right there. Now AUG has this funky notation start, and then it also has an amino acid by the name of methionine. Now this is a little bit funky, but I'm going to go ahead and write methionine, which is abbreviated MET. And I'm going to delay talking about what start means and go to the next one. So our next one is going to be AUC. Okay, I see AUC here, and AUC maps onto an amino acid named isoleucine. Which gets the abbreviation ILE. You don't need to know those abbreviations, but you do see them, like in the previous image, 3 letters representing a specific amino acid. So I just want you to be aware that there is another way to represent amino acids other than giving you their full name. Now let's go ahead and see if we can do the last one. Why don't you see if you can find this one? Where is UCG on this codon chart? And it might take you a little while, but hopefully you found it up here. And UCG codes for serine. Now, real quickly, what do these amino acids look like? Well, again, we've already learned that amino acids are classified based on being polar or nonpolar or electrically charged. And for this exam, if you figure out what a particular sequence is, like this one, methionine, isoleucine, and serine, you are expected to also be able to look at the structures of those amino acids if provided, and determine if those amino acids are polar, nonpolar, or electrically charged. I'll go ahead and draw this out real fast. That's my backbone, and now I'm going to go ahead and draw in the residues for each one of these. Okay, so based on this, I think it would be pretty obvious that because this one which is isoleucine, has nothing but carbons and hydrogens in this. This one is going to be nonpolar. And this one over here, serine, has an OH in it, so that makes it polar. Now methionine is a little funky because there is a sulfur in methionine, but otherwise it has carbons and hydrogens. Sulfur is not nearly as electronegative as oxygen is, so technically this one is also nonpolar. So if you know what the designations are of the various amino acids in terms of are they polar, nonpolar, electrically charged, you can predict what they might do when they get ready to fold into their 3D conformation. And we'll explore that in more detail in a little bit. Now that you've had an opportunity to see the steps in action, it is your turn to give this some practice. In this image, I have a double-stranded DNA molecule. I have told you which one is the template strand. You ignore the black strand and you only focus on the blue strand. Again, I'm showing it to you in this way because DNA is truly double-stranded. However, to make things simpler on an exam, I will only give you the template strand. You might notice that I have these markings here called 5' and 3'. Those give you the direction of that DNA. And it's a little bit beyond our scope, but it technically provides some reference like start and end to the DNA molecule. So you're going to go ahead and run your RNA transcription in the direction of this arrow that I've shown here. And when you go ahead and start, you're actually going to start with what's called a 5', and then you're going to end over here with the 3' of RNA. In other words, 5' and 3' always run in opposite directions to each other whenever you are doing this base pairing setup, whether it's two strands of DNA or DNA to RNA. And if that doesn't make a whole lot of sense, don't worry about it right now. Go ahead and just try the transcription and then use that transcription to go ahead and figure out the amino acids. Pause the video and go ahead and try on your own. All right, hopefully you gave it a shot. Now I'm going to go ahead and do it as well, just to make sure that you have the right answer. And if you don't, maybe you can figure out what went awry and then learn from that. So we are going to create an mRNA transcript going this way. So remember, just up here in my little corner, I'm going to put my DNA and my RNA, which you need to memorize. All right, so now that I have that, let's go ahead and make this RNA transcript. So T is going to pair with A is going to pair with U, C is going to pair with G is going to pair with C, etc. So let me go ahead and finish this real quick. Okay, now what we're going to do is we are going to break this up into groups of 3 so that I can identify the codons. So let me go ahead and underline the codons. And so I can use this to figure out what amino acids I have. So again, I start with AUG, which seems to be the start thing that I keep starting with. So that's going to be methionine. Now I'm going to link methionine to whatever CUC codes for. CUC codes for leucine. And now UUU. Come up here, UUU codes for phenylalanine. Now I have GGA. That's glycine. Now I have AUC. That's going to be isoleucine. And then finally I have UAA, which codes for stop. So if you got this, great job. Let's go ahead and talk about those start and stop codons. All translation starts with AUG, and therefore the first amino acid in any translation is going to be methionine. For this reason, it is called the start codon. Now, are all proteins built to start with methionine? Not necessarily. Sometimes you can go and cut off methionine after you've finished your translation, but it always seems to start the process. So just because you start with that doesn't mean you'll keep it in your final product. Now, what about these stop codons? So for reasons that are beyond the scope of this discussion, there's usually overhang on the RNA, meaning there's additional RNA beyond what you actually want to translate. So if you have RNA that looks like this, here's my code of RNA. Here's all of my bases using that train track designation again. The start codon might be here, and the stop codon might be here, which means that this is the only region that actually gets translated. Everything else, this is all extra. Again, I'm not going to talk about why it's extra, but there is usually extra left over. So the stop codon basically tells your ribosome, and here's your ribosome that's on it, to basically stop doing translation and come apart. And when that happens, translation is over, and the rest of this should not be translated. So stop codons are not actually amino acids. So technically, I really shouldn't have this here at all. It should just be the end, because stop is not an amino acid. But we need to know when the translation stops in our sequence. So that's why they are important. So the last thing that I want to have you do is go ahead and take a look at the residues for the amino acids in this chain. Methionine, leucine, phenylalanine, glycine, isoleucine. I mean, you can go ahead and pull up an amino acid chart and take a look at them. And what you will see is that they are all nonpolar. So that tells us something about how this chain might fold on itself. The fact that they are all nonpolar means that those residues, if surrounded by water, are going to want to come together, and that will help the whole thing fold in using hydrophobic interactions. All right, that concludes our lecture. Thanks for watching.